Okay, thank you very much for being here. So my name is Djamel. I'm from chemistry department. I work there as an instructor and a research scientist. Unfortunately, my colleague Dave Hasman is not here today. He's attending a conference in the United States. So I'm doing everything. Yeah. So I'm going to talk to you today about some anti-cancer compounds. They are called polyacethylenes. And I think the laser pointer is not working with this. So the type of cancer that we are dealing with in this project is pancreatic cancer. It is one of the worst types of cancer. So that's why actually discovery of new compounds, new treatments is an urgent matter. So the objectives of this project was actually to look for this compound or to identify this compound by chemical extractions, organic synthesis, and then test these compounds for anti-cancer activities, including like anti-proliferation properties. So before we get into this, like chemical extractions, organic synthesis, bioassays, so first I'd like to introduce you to this type of compounds that we are investigating here. They are called polyacethylenes. So what is a polyacetylene? Polyacethylenes are organic compounds. If you are new to organic chemistry, so organic chemistry deals with carbon compounds. So organic compounds are carbon compounds. Like for instance here, so this is the structure of one of the polyacethylenes. So there are 18 carbon atoms. So one of the most interesting properties in terms of structure of these compounds is the presence of triple bonds. So in organic compounds, there are three types of bonds, like single bonds, double bonds, and triple bonds. So this is a triple bond here. So a triple bond between carbon 4 and 5, carbon 6 and 7. So we have two triple bonds. So if a molecule like this has two or more triple bonds, then we can say that this compound belongs to the polyacethylenes family or polyenes family. So now you have an idea about the polyacethylenes. Other thing, so the presence, so apparently the presence of triple bonds, that's what makes this compounds to have anti-cancer properties. So in the compounds we are dealing with here, so we have only two triple bonds. So this compound, this is actually was the first polyacetylene or the first compound that we investigated in this project. So this compound was extracted from Devil's Club. I don't know, probably you heard about this. So this is, so I'm going to use here the abbreviation DC. So Devil's Club is a local medicinal plant in BC. It's been used for probably thousands of years by the First Nations to treat different diseases and also for religious purposes. So the Devil's Club plant is an important medicinal plant. So here I'm using the name or so this is the actual name, like the scientific name or chemical name for this compound. It's a long one. So I'm just going to use an abbreviation here. So the abbreviation here is DCA. DCA, so that means this compound is extracted from Devil's Club. A, this is due to the presence of a group here. This is a group called the acetate. So that's why we are using this common name or an abbreviation to name this compound. Because I'm going to show you other structures a little bit later. So we will be using different abbreviations to name this compound. So this very compound that I just showed you, so it is a polyacetylene, and then it was extracted by my colleague Dave Hasman a few years ago. He's from forensic sciences. So the extraction process involves, so he collected the plant from somewhere in BC, probably North Vancouver. So, and then the next step, so involves drying, grinding, extraction with organic solvents. I think he used here methanol, he used ethanol and water. So the next step was chemical analysis and then bioassays experiments. So bioassay experiments, this is when this compound was tested for anti-cancer properties, and then it was found to have interesting or important bioactivity, so including anti-proliferation and then also other anti-cancer properties. So now, having said that, so we have an idea about the structure of DCA, so it's a polyacetylene, and also we have an idea about the activity of this compound. So the next objective, what we want to do, we want to synthesize compounds and then do the same. So analyze and then test them. And this is my job here. So I am here. So I'm here to synthesize this compound and again analyze them and test them. So before actually I go to the synthesis part project, so I have it's very important to mention that these compounds, like these polyacetylenes, so some of these compounds they have, in addition to these triple bonds, they have, some of them have a single bond here and others have a double bond. So I thought, like for the sake of simplicity, so I looked for this compound. So I did some research. I thought it would be like easier or simpler to synthesize a compound with a single bond here. So this compound is called 1,2-dihydrofalcarinol, and I'm going to give it the abbreviation FCH. I really need my, I miss my laser pointer. Okay, so to make this compound, so I need to synthesize or prepare, or two small molecules are required to synthesize this compound here. So the first compound is this. So it's called the chloro, I'm just going to call it the chloro compound. And then the second compound here, this is which will give me the polyacetylene parts. So I synthesize this in 2 steps, and then I synthesize this compound in 1 step, and then couple the two molecules together, and then I prepared this polyacetylene now, which I'm going to call FCH. So I did this for the first time, so I've never synthesized this before. So this actually was a very good starting point. So at this point here, so we proved 2 things. First, we have the expertise, the equipment, the labs to synthesize these compounds, because we confirmed the structure of this compound. And then when we submitted a sample of this compound to UBC and VGH, so they said it is active, but it's not as active as DCA. So that's the difference. So when they told me this, so I looked at the structure of DCA. It has a double bond in this position here. So I thought it's a good idea to synthesize this compound with a double bond. So that was my next target. So that was my, my second target, second molecule, so with a double bond here. So to make this compound, so I needed the chloro compound, which I've already synthesized before, and then this second molecule here with a double bond. So I just need another reagent, or a different reagent, to make this molecule here with a double bond in here. So again, I applied that reaction. So I reacted the 2 molecules, and then I got this compound here. See, this is called FC, or falcarinol. This actually compound, it's known, like it's been synthesized before. And also, it is a very interesting compound because it has different bioactivity properties. So for instance, it is an antioxidant, antibacterial, antiviral, so it is very active. So what's unique about this compound, so nobody has used it to treat pancreatic cancer. So we are the only group who's going to use this compound. So when I submitted this compound for bioassays experiment, so actually they usually send me an email, but this time they called me. They said, this is the one. Yeah. So it was like, so it was more active than FCH, and then it was more active than DCA. So that was very interesting. It's a very interesting compound. So So before I come back to the bioactivity, let me just first give you a summary of the compounds that we have identified here at BCIT. So remember, the first compound was DCA, so that was extracted. And then this is the second compound, which I synthesized with a single bond here, and then FC with a double bond here. And then actually I managed to make another analog of FC, so by introducing a group here, so I called it FCA. So far here at BCIT, we have identified 4 anti-cancer compounds, or 4 bioactive compounds, but the best one, or the most effective one, is this one here, FC. So all the compounds that we have synthesized, final products and intermediates, so all of them were characterized by different spectroscopic techniques, different chromatographic techniques, including LCMS. So we analyzed our compound by LCMS. This is, LCMS stands for liquid chromatography mass spectrometry. We have a very sophisticated equipment, or LCMS, here at BCIT. So one of the outcomes from this project was that when we analyzed the DC extract by LCMS, and then we analyzed the synthesized compounds, so we proved for the first time that these compounds, so FC and FCH, which I synthesized, they are also produced by the DC plant. So from the chemistry point of view, this is a very interesting result. We have submitted a paper, and it should come out very soon. So this is the LCMS analysis. So I'm not going to go over this in details. So these are here the LCMS chromatograms of the synthesized compounds. These ones here are for the extracted compounds. So we proved that FCH, this is with a single bond, was present in the extracted compound, and then also FC was present in there as well. So this is in terms of the chromatographic analysis. The mass spectra also are identical. So this is the mass spectrum. So I didn't label it. So this is the mass spectrum of FC, and this is in synthesized. This is the extracted. They are exactly the same. So well, we were actually the first group to report this. So especially by analysis with by LCMS. Sorry. So coming back to the bioactivity. So they've done a lot of work actually at VGH and UBC. I'm just going to show you a sample, so it's not my area. So this is the cancer cells before treatment. This is after treatment using FC, that's the best compound, at 100 nanograms. So 200 nanograms, and then the dose was increased to 500 nanograms. So as you can see here, in the last diagram, so most of the cancer cells were detached, they decreased in size, and then they just died. So it's really very interesting. So probably I should get someone from UBC or VGH to talk about this, because they've done a lot of work. So I'm just giving you, I'm showing you here a sample. The other conclusion, remember when I mentioned double bonds versus single bonds. So one of the outcomes of this project was that we found out that the compounds with double bonds in this area, so in addition to the triple bonds, so with double bonds here versus single bonds, these compounds are more active than the compounds which have a single bond. So this was reported by other groups, but again, we are confirming these findings. Another conclusion, another outcome from this project, so FC, that's the best compound, is actually present or produced by carrots. So when another group at another university, so when they tried to extract FC, they had to use 10 kilograms of carrots and a large amount of our organic solvents. to extract 10 milligrams of these active compounds. So whereas here in the lab now, I can use a small-scale reaction. So even my student did it last week. He's here, Anthony. So a small-scale reaction. So actually, we can make grams. We can scale it up. So chemical synthesis may provide here an advantage over the extraction process. So that's another outcome. So far, all the experiments that have been carried out at UBC and VGH, they were carried out in vitro, like where they had cancer cells in Petri dishes. So the next step, they are going to carry out in vivo experiments, using animal models, and that's what we are doing now at the moment. We are going to make actually more compounds or more FC, larger quantities for preclinical testing and also to carry out this in vivo experiment. So remember I said that FC is the most effective compounds. At the moment, as we speak during the summer, we are trying now to make new analogs, including, like I'm trying to introduce new groups to the FC compound. And also, we are thinking about probably synthesizing compounds, or these compounds, with a triple bond. Remember, we have single bond, double bond. We are also planning to synthesize triple bonds and test the activity. We have a poster. So if you have, so Anthony is working with me this summer. So if you have any questions, you can actually ask. I summarized all this presentation in one poster. So I'd like to thank the Vancouver Hospital and also the Hecht Foundation from Vancouver, and also BCIT Research, and also our School of Computing Academic Studies for supporting this project. I'd like to thank all my colleagues from chemistry, forensics, physics, and basic health sciences for their technical support. I'd like to thank also the research group. So Dr. Dave Hasman from forensics, so he took care of the LCMS analysis. Susan Chung and Joe Tai, and they have also a large research group at UBC for bioassays experiment. We also work with Dr. Richard Smith from University of Waterloo. He has like a mass spectroscopy facility. He also helped us with the LCMS analysis. I'd like to thank all my students. So again, I think it's very important to get the students involved. So I had a number of students working with me for the last few years. So including Anthony, he's here. Maybe you should stand up and say hi. Yeah. He's doing a good job. Thank you very much. I don't know if you have any questions. Do we still have a few minutes for questions? Or yeah, okay. Thank you. So this project actually involves extraction, synthesis, LCMS analysis, and then bioassays experiments. It's actually a lot of work. So my area is organic chemistry, organic synthesis. But if you have other questions about other areas, I'll try my best. Yes? Do you have any idea or any clues about the mechanism that works by at this point? Or is that like for the anti-cancer? Yeah, I'm not sure. All I know, actually, so this compound, the FC compound, they've done more work. So it's not only active against pancreatic cancer, it is active against all cancers. So brain cancer, liver cancer, all colon cancers. So I think, I'm not sure about the mechanism, but it's very active. And also like one of the, what I hear from my colleagues, so actually it does attack only cancer cells. I don't know what the term, but it doesn't touch the normal cells at a certain concentrations. But yeah, maybe we should get someone from UBC maybe next year and VGH, my colleagues, to talk about this project. Yeah. Yeah.
So what was the efficacy of the analog compared to the original FC?
No, we haven't tried yet. No. Yeah. So we are working now. So Anthony is helping me with that. So no, not yet. So we have to confirm. So that's what happens. We synthesize it, analyze it. Then when we are confident about the structure, then we submit a sample for bioassay experiment. So we are not at that stage yet. Oh, the other analogs. Oh, okay. Actually, the last one I synthesized, the FCA, actually it's just as good as FC. Yeah, it's just as good. So, but so far, I don't know why they choose to use FC, like the farcarinol. The other one with a single bond is less active, but I'm hearing that it's good against brain cancer. That's why the mechanism is really, I have no idea. So all those 4 compounds are active. So FC is the best. FCA, which I introduced a group in the OH, so it's just as good. Yeah. Yeah. Oh, I think they grind. So they use it as a tea. I think like make tea and a solution. And also I think they dry it and then mix it with berries. I think they use it like against lice, and also, but they used it against different diseases, different it's good for diabetes. So I think our group at UBC, they are trying to communicate with the First Nation to tell them how important is that plant. So they are doing that. Yeah, so it's a good question. Yeah.
But with a much more altruistic angle. Sure. I just have a question about what you had synthesized in terms of its stability and its shelf life. What is it?
It's a good question. So these polyacetylines, so remember we said that if you have 2 triple bonds or more, then the compound belongs to this class of polyacetylines or polyenes. So the more you have triple bonds, like if you have 3 or 4, then it's light sensitive, so it will decompose quickly. So I think we are lucky with these compounds. We have only 2 triple bonds, so they are actually quite stable. It depends. So it might last for a year or something, but so far you can ask Anthony. I always ask him, so make sure you store it under nitrogen, like we use nitrogen atmosphere, seal it, and then minus in the freezer. So I think I'm thinking of actually getting a minus 80 degrees freezer. So it's a good question. Yeah, they are not very stable, but they can last definitely for a few months, definitely. Yeah.
So I'd like to thank Dr. Khelifi for his presentation.